Thank you very much for awarding the Fellowship of Indian Academy of Neuroscience, which I have been associated for the last 30 years. And also with this, I'm hoping that the neuroscience between India and Sri Lanka will gather momentum, and we would like to work together for the human neuroscience. Thank you.
Thank you so much to the organizers. Over.
Thank you, sir. And on behalf of the organizing committee, I would now like to congratulate all the newly elected fellows of the society. So now it's a great privilege to have Dr. Prahlad K. Seth as a guest of honor for this morning's opening session of IAN 2021. Dr. Seth is the former director of Industrial Toxicology Research Center and Chief Executive Officer of Biotechnology Park, Lucknow. He has made notable contributions to molecular biology and biotechnology related to health sciences, particularly understanding the effect of chemicals and drugs on the nervous system. He introduced the concept of mechanistic toxicology for assessing the risk to toxic substances and the need to identify target molecules to serve as markers of exposure and prediction of adverse effects of chemicals. He is a member of several committees and has received numerous awards and honors in his career. He is an elected fellow of the National Academy of Sciences and the National Academy of Medical Sciences, India. He is the founder fellow of the Indian Academy of Neurosciences and has been the president of the Academy and several other organizations. I would like to request you, sir, to kindly deliver your speech as a guest of honor.
Thank you very much, Maua, for your kind words. Indeed, it's a great pleasure to be talking on this occasion when we are having the 39th annual meeting of the Academy. Professor Bruce Alberts, who is the chief guest of today's function, Professor P.N. Tandon, the eminent neurosurgeon and neuroscientist who had played an important role in the growth of the neurosciences in the country, Dr. Raju, the Dean of the Academy, Dr. Ishan Patro, President of the Academy, Dr. Vinay Khanna, Secretary Headquarters, Dr. Pankaj Seth, General Secretary, the Dynamic Organizing Secretary, Dr. Jayashree Das, and the two joint organizers, Dr. Biswas and Dr. Ghosh. I think I would just like to mention about, people have talked about the Academy, that how and why the Academy was formed. In fact, the idea of having a platform for the neuroscientists from various disciplines was mooted by late Professor K.P. Bhargava, an eminent neuropharmacologist of the country. And he mentioned about and gathered the neuroscientists. And it was realized that, in fact, there is a Society of Neuroscience already exists. And then it was felt, okay, let us have a conference of the Neuroscience Society. And Dr. Mehdi Hassan, the late Professor Mehdi Hassan, who had done remarkable work on the neuroanatomy, volunteered to hold this meeting in Aligarh. Invitation was sent to the then president of the Society of Neuroscience India, late Professor Ramamurthy, and the General Secretary, Dr. Subbarao. And with their permission, the meeting was organized, but unfortunately, they were not able to come and participate. Professor S.S. Parmar, who was visiting the Aligarh, and this was decided to have a body, and it was felt that we should have because there's a large number of the neuroscientists working in clinical areas and the basic sciences and allied areas, a platform should be there. So in 1983, when this meeting was organized, it was felt that we should have, and it was named as the Indian Academy of the Neurosciences. So that's how the Indian Academy of Neuroscience is born. And then we had got it registered in 1984. With these stalwarts and the another person who played a very important role, late Professor B.N. Dhawan, who was really an architect for getting people there and organizing and guiding, and a lot of work he had done for the academy. So finally it got registered in 1984 in Lucknow, and then it kept going on, and the academy has rose. And then we had blessings of eminent neuroscientists like Professor B.K. Bachawat, Professor P.N. Tandon, who accepted our request to be the president of the society, and they guided us. The academy soon developed international linkages, and thanks to Dr. Vijayalakshmi Ravindranath.
Thank you.
That was for the FONS, and
Thank
this is the Federation of Asian Oceanic Neurosciences, and also the EBRO. And then it has linkages with many other, as was mentioned by Dr. Pankaj Seth, that with the Japanese neurosciences and many, many other formal and informal linkages, and we get representation from there. Academy has number of chapters, and these chapters, one of the chapters in USA also, which was founded by Dr. Subhash Pandey and Dr. Ghanshyam Pandey, and they also have been very, very actively providing the guidance and in the growth of this. The academy has a tradition of organizing a theme-oriented conference along with the international representation of the society of the scientists from all over the world, and that has been going on. It has instituted a number of awards to honor the scientists who are working, and also in name of the well-known and established scientists of the country. This meeting, which is happening in Calcutta, I must pay my tributes to Professor J.J. Ghosh, who was one of the pioneers of starting the neurosciences, and particularly in the basic sciences at the Calcutta University. Dr. Ghosh has mooted the idea of having a brain research association, and I think that was one. So still, as we see, India is a large country, and there is a need of having many, many more neuroscientists coming together and using a platform where they can exchange the idea, where they can provide. The neuroscience, as we see, is going to play a very important role. It has played an important role, and there are many, many areas in which still we need to work, and that is like this COVID. What is the COVID, lingering COVID, brain fog? Still, we do not know, and that happens with the many neuroinfections that is needed. The India, like other world, is aging. More and more aged population are going to be there in the country, and how we can work and find solutions for a better life in the aged population in the India. The diseases, the neurodegenerative diseases are going to increase more and more. So we are about 1,500 plus members in the Indian Academy of Neurosciences, and yet the number is very small. And so still I will say we have a little critical mass, not a real critical mass, which can work and obtain and help in the solving the problems of the neurosciences. The Department of Science and Technology, Department of Biotechnology are providing support in the area of the neurosciences, and so is being done by the Indian Council of Medical Research. The contributions of these stalwarts, I think we owe a great respect to them, and what they have contributed in the growth of neurosciences. And I see the new teams which are coming together, and like the Dr. Jayashree, who has organized this conference, it's a wonderful meeting which is going to be I'm sure is going to be another feather in the cap of the neurosciences. So with this, I will close, and I once again thank the opportunity which has been provided to me to be talking to you. I should not be a guest of honor. I am a part of the Indian Academy of Neurosciences, but anyway, nevertheless, I thank you once again for giving me this opportunity. Over to you, Mahua.
Thank you, sir. We are really honored by your presence in this event. And now I would request President IAN, Dr. Ishan Patro from Jivaji University, Gwalior, Madhya Pradesh, to chair the inaugural session.
Good morning, Dr. Bruce Alberts, Professor Tannen, Professor Seth, all the members of the executive committee, delegates, ladies and gentlemen. I have the great pleasure of introducing Professor Bruce Alberts, who will be delivering the inaugural lecture today on the topic, the great complexity of biology from cells to tissues. Professor Bruce Alberts is in the Department of Biophysics and Biochemistry, University of California, San Francisco. He's one of the prominent biochemists we have in this world. He served 2 6-year terms from 1993 to 2005 as the president of the National Academy of Sciences, USA. He has done significant work studying the protein complexes that enable chromosomes replication during cell division. One sentence introduction will be enough for all biologists sitting over here, that no biologist reads biology without reading the book they call the Bible of Cell Biology, The Molecular Biology of the Cell, of which he is the principal author. His strong commitment to improve science and mathematics education is commendable. President Barack Obama honored Dr. Bruce Alberts with the National Medal of Science Award in 2014 and the Lasker Special Achievement Award in Medical Sciences in 2016. Dr. Alberts served as editor-in-chief of the magazine Science during 2009 to 13, and as one of the first 3 United States Service envoys during 2009 to 2011. During his tenure as the president of the National Academy of Sciences, Bruce was instrumental in developing the landmark National Science Education Standards that was implemented in schools system nationwide. From 2000 to 2009, he served as the co-chair of the Inter-Academy Council, a new organization governed by the presidents of 15 national academies of sciences and established to provide scientific advice to the world, committed to the international work to the promotion of creativity, openness, and tolerance that are inherent to science. Alberts believes that scientists all around the world must now band together to help create more rational, scientifically based societies that find dogmatism intolerable. Widely recognized for his work in biochemistry and molecular biology, Bruce has earned many honors and awards, including 16 honorary degrees. He currently serves as advisory boards of more than 25 non-profit institutions, including the Golden Betty Moore Foundation. While we requested him to write the foreword for our book, which is going to be released today, Bruce writes, India, a wonderful country that I have visited perhaps 20 times, has an immense scientific and technical potential. We are really indebted to you, Bruce, for accepting our request to be here and delivering your lecture, and also to accept the honorary fellowship of the Academy of Neurosciences. It's over to Professor Bruce Alberts for your lecture, sir. Bruce, would you please unmute? Bruce, please unmute.
Sorry. It's a pleasure to be here. It's late at night in California. I'm going to try to share my screen. I hope this works. Let's see, I gotta get this. Well, I gotta try again. Well, there we go. I got well, I gotta go back, but there we go. All this technology is too much. So this is a great pleasure for me to be able to be here virtually and see some of my friends in India. I haven't been able to travel for 2 years. Maybe next year I'll get there again. I am not a neuroscientist. I'm a cell biologist and biochemist. And so I'm going to talk about science that's relevant to neuroscience, but I'm not going to tell you anything about neurosciences per se. This is an outline of my talk, really is based on my life. If you add up these numbers of years, you see I would be 105 years old. So these periods overlap. But I spent 35 years as a research scientist. And then I started writing textbooks. And then I became president of the National Academy of Sciences, and I've been working on science policy. So I'm going to cover all those grounds. I'll try in 30 minutes. So I worked on the biochemistry of protein machines, and I think it was a wonderful, a lot of fun. I started thinking about science in high school, and at that time, although I didn't know it, the Watson and Crick famous structure of DNA was proposed, and it made a revolution in our way of thinking about doing molecular biology and molecular science. They solved the mystery of heredity in theory, but they didn't know anything about the machinery that enables a DNA sequence to be copied from one generation to the next. And so starting as a graduate student, I would spend about 30 years trying to work on this problem. And it's really been an enormous change in how we conceptualize how cells work. When I started in 1957, we thought of a cell as, a test tube full of a random mixture of highly concentrated protein and RNA molecules and small molecules, and that the collision rate was so fast between them that everything was bouncing around. And so DNA would be replicated by this enzyme that had just been discovered, DNA polymerase, which won a Nobel Prize for Arthur Kornberg. It would be replicated by this one enzyme, and it would, add a nucleotide as Watson and Crick predicted, and then fall off, and another one would rapidly collide, and everything would be based on random collisions in a randomly mixed up cell. So I had a very dismal career as a graduate student. Students love to hear about this. I had my own theory about how DNA polymerase might be able to replicate DNA, even though the DNA helix was double-stranded, and the polymerase in the test tube would not work on double-stranded DNA. And I did many experiments trying to test my theory, and of course it wasn't correct. And it was a very bad way of doing science. Nevertheless, I thought my professor after 5 years said I had done enough and I should go on and get my PhD. However, I had an enormous surprise. I walked in these a group of famous Harvard professors, and they told me that I didn't pass and that I would have to do more experiments before I could get a PhD. And that, as I explained in this little paper in Nature, it was a very important experience in my life, because in this case and in many other cases, you really learn from failure. You have to really focus on what went wrong. And I spent about a month thinking about how I had not done science correctly and how I do much better in the future. So the first thing I learned was the crucial role of strategy in science. being a good scientist is like being a great artist. I like to talk about Rembrandt painting paint and pictures. You could have all the paints that I could have all the paints that Rembrandt had and everything, but I wouldn't make anything good out of it. Science is a creative endeavor, and it's much of the real fun of it is thinking through a good strategy for your research that's different from whatever people are doing, but also has a real good chance of finding out something important. I had not done that. I had a very bad strategy in my as a graduate student. Finally, I did get my PhD. I moved to Geneva in 1965 as a postdoctoral fellow, and I immediately discovered that my whole hypothesis was wrong, because genetics had shown that DNA is not replicated by just DNA polymerase, but by a whole set of different proteins. And I discovered there the bacteriophage T4, a wonderful model organism for the early days of molecular biology. And it makes all its own DNA replication enzymes. And genetics had been done and published, while I was in the middle of my thesis, but I never saw this paper. Genetics had showed that many different proteins are needed to replicate DNA in addition to DNA polymerase, a total of at least 7 proteins. And so another basic lesson is much easier today because of the internet. Really read widely so you know what's going on and can make a good strategy instead of a bad one. So after a 1-year postdoc in Geneva, I returned to become an young assistant professor at Princeton University, where I'd be for 10 years. And I had graduate students and undergraduates, and we focused on purifying each of the bacteriophage T4 proteins that have been identified by genetics, with the hope that someday we'd have 7 purified proteins. We could mix them together with double-stranded DNA, and the DNA would replicate. It took, as you see, 11 years. Finally happened in 1975. And what we and others, obviously working at the same time, discovered was that that DNA is replicated by a protein machine. In the case of T4, 7 different proteins. 2 DNA polymerases are part of this machine, one going forward and one going backwards, and they're linked together to form this little loop. It's just amazingly elegant way of making DNA that nobody would have predicted, that had to be discovered by experiments. So I always like to show this favorite movie, which shows DNA being replicated in a pretty realistic way, moving at about the same rate as the DNA is replicated in human cells. This is a little video that was made on the 50th year of celebration of the discovery of the DNA helix.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Well, this chemistry is just typical of what makes life possible. There are thousands of different protein machines, all working differently, but that's, this shows the great sophistication of life's chemistry. So the basic lessons, I think, are because of evolution, there are remarkable homologies between living things. Therefore, use model organisms wherever possible. We use this T4 bacteriophage, which was the same mechanism as human cells, but much simpler. And in understanding those simpler systems, then there's a shortcut to understanding humans. And secondly, nearly all cell processes will be driven by a protein machine. That is, by 10 to 20 proteins organized in a complex and incorporated ordered protein movements driven by the energy of ATP hydrolysis. You need to have directional motion, and that's why we have electricity to drive our machines we know in everyday life. And this is done by mechanisms of allosteric changes driven in one direction by the hydrolysis of very tightly bound ATP. And finally, everything in life is going to be based on elegant mechanisms that are too complex to predict. They have to be discovered by experiments. And so one challenge for the next generation of biochemists is doing this kind of work that I and others did, purifying proteins and reconstituting protein machines so we could really understand how they work. And there's a lot of that still needs to be done. Many machines like the ribosome, we know how it works, but we don't know how many of the other machines work. So now I'm going to switch to 40 years of textbook writing, which taught me that we're far from understanding biological complexity. There's a huge amount of work for young scientists to do all around the world. I started writing a textbook because Jim Watson had this brilliant idea. It was time to bring together molecular biology and cell biology, which at that point were two different fields. He got this group of authors together and said that it would only take us 2 summers. But of course, that was not right. It took over a year of days working together to write this first edition of this textbook. Here's the first set of authors. Jim Watson's back there in the hat. He's no longer 25 years old. He's now like 55 at this point. And here's the book that's coming out in another month or so. This is the 7th edition of our textbook. And the main thing I want to point out is each time we write a new edition, we're humbled by how much we still don't know. So here's 2 surprises of many that we learned in the last few years. The recognition, first of all, that everything's organized inside the cell by scaffolds and condensates. That is, they're membrane-free organizations that bring biochemical components together so that they could function together. And the second thing is that that life is just so complicated that almost everything that really want to understand is what physicists call an emergent property. You can't just understand it from knowing the network of interactions. We need to be able to use math and computer science to somehow get meaning out of this great complexity. So I'll go through both very briefly. Scaffold proteins, here's a little diagram from our textbook. They bind specific other molecules like proteins and cause them to collide very rapidly so that the reaction is now much faster than it would be without them. And also they are located in specific regions of the cell, so they make these reactions happen where they're most needed. The simple example, an early one, was the polymerase of actin, which the actin filament is a major component of the cytoskeleton. So this red thing would go all the way down through the floor. And this is the growing tip of it, catalyzed by a protein called formin. And it grows faster than the diffusion control rate, which would be impossible, except that you have this little scaffold bringing the red little actin monomers into close approximation to the end so they could collide very rapidly, and making the reaction much more efficient. Here's a much more complicated kinds of scaffolds. You look inside of a cell nucleus. When DNA is being replicated, you get a concentration of proteins that people refer to as replication factories. You have transcription factories. Those things come and go like these condensates only when they're needed. And here's a bid to neurosciences. I am just fascinated by these condensates that form neuronal granules, the molecules that bind selected messenger RNAs and carry them out to synapses, both in dendrites and axons. And there's all kinds of mysteries there that we don't understand, but very important for understanding neuroscience. Here's a recent review that I think would be very useful for people who want to get acquainted with that aspect of neuroscience. In conclusion, a cell is nothing like a test tube. Nearly everything is organized inside the cell by protein and RNA scaffolds. Emergent properties. Okay, this is a very quick introduction to what I am talking about. Basically, when I was a student, we had this this standard thing: that A goes to B, goes to C, goes to D, and those pathways really wouldn't work inside a cell without feedback and feed-forward networks that control them. And even these relatively simple things require mathematics to understand them. But of course, the cell is much more complicated. Here's the actin filament network in a yeast cell, so it's simpler than a human cell. These are all the proteins that bind to that actin filament, the major protein of the cytoskel, to make it do the right thing at the right time. They're proteins. The purple ones are like that formin and control filament dynamics, and they're the myosins, the famous motor proteins, the green ones, and all these things somehow in the cell make organized structures in the right place at the right time, and we just don't understand that. It's just too complicated. Even when you know everything, you can't really make sense of it. So here's a little nice review, again, another recent paper about biological feedback control and all the loops that need to be understood through sophisticated mathematics, that some of which has not been really developed yet, and new ways of computationally dealing with this stuff, and new kinds of experiments are very badly needed. So as a consequence of such complexity, even when we have a complete knowledge of the molecules, protein machines, and molecular interactions in the cell, we're not be able to understand even the simplest living cells. Instead, life reflects the emergent properties that result from very complex networks of chemical interactions. And that's great for young scientists. There's so much to do. My friends think I'm way too optimistic here, saying it will take the rest of the century to get a real understanding of how cells and organisms work. We're going to need all kinds of new research and a lot of energy. So now I'm going to end with my 30 years of science policy, which I want to make just the statement that wise investing for the future demands much more support for the fundamental biological sciences, which are needed to get us to understandings of life that we badly need to do anything we want to do in more applied research. To me, a current problem is that the major funding agency in the United States for biological research has been overemphasizing shorter-term translational biomedical research, which of course is important. But with so many unknowns and so little understanding, fundamental research on biological mechanisms also remains absolutely critical for improving human health. In my favorite editorial, I was editor-in-chief of Science Magazine for 5 years, and I commissioned something like 250 editorials. This is my favorite one by Huda Zogby, who's a great translational neuroscientist in Baylor. She was an MD working on Rett syndrome and couldn't help these children. So she decided she'd go into research, and she's just been a fantastic translational researcher. But she wrote this editorial for Science, pointing out that too many highly qualified basic scientists are being pushed into translational medicine. We need more, she says, we need much more fundamental research. And she really talks about herself having to work with the biological system that's sophisticated, like a text by Shakespeare, using a first grader's vocabulary and experience. I just love that quote, right? Because she's from Lebanon, and so of course she obviously knows the difficulty of translating between languages. So her point is, in order to really be able to translate, understand the cell, we need much more fundamental research. Just one example, the importance of the fruit fly Drosophila. We're still far from understanding how cells work together to form and maintain tissues. Many examples show that the first working out a mechanism in the fruit fly provides a shortcut to understanding humans. And this is an example in our developmental biology chapter. In the last edition, we had 50 references to Drosophila, 4 times as more than the next most cited organism, which is the mouse. You know, without the breakthrough findings in Drosophila about developmental biology, we still wouldn't understand much about humans. So just to end with the human brain, I really admire people who work on it. It's so complicated. The ultimate emergent property is human consciousness. It's a property that comes out of the network of nerve cells, and it's quite different from the things that make it up. So to understand what the physicists meant by emergent properties, just think about human consciousness coming out of a network of nerve cells. Similarly, at the cell level, the cell somehow knows how to think, coming out of a network of interactions. And you all know this, the human brain is incredibly complicated. And so the Drosophila brain contains about 100,000 neurons, 1 million times less than humans. And you have all these advantages with Drosophila. You could make mutants, you could trace every neuron, and so on. And I just think there's no chance of understanding the human brain until we understand the Drosophila brain. This is an example of where we need much more focus. So my major point is that, as Huda Zogby emphasized in her editorial, the many scientists who are working to improve human health will make much more rapid progress once we gain a better understanding of the chemistry of living cells and tissues. And thus, the fundamental research needed to gain this understanding is an essential investment for humanity's future. I just want to end with this slide taken in about 20 years ago, a meeting of 20 academy presidents in Geneva, in Switzerland, in Davos, Switzerland. And here we are at a Swiss McDonald's coming home. And this is about 1/4 of the academy presidents, but it happened to have both the Indian Academy presidents, Prakash Tandon, speaking next, and Govardhan Mehta, both of whom became my close friends. And right behind Prakash is the president of the Chinese Academy of Sciences, and then the Japanese Academy. So thank you very much. It's been a pleasure to be able to talk to you.
Thank you, Bruce. Now I would request you to formally announce the launching of the book, The Biology of Glial Cells, Recent Advances, published by the Springer Nature. And once you announce the launching, Dr. Bhavik will play a slide presentation, and he'll speak about the book also. Hello, Bruce, do you listen to me?
Hello?
Bruce, this is
Yes.
Isam. I would request you to formally announce the launching of the book.
Okay, all right.
Yeah.
All right. I've never done this virtually. I've done it in person several times in India. I am very pleased to launch this book about glial cell biology. It represents a major contribution by, I think it's 25 different articles. Many of the people presumably at this conference were contributors. I know how hard it is to write a book and how wonderful it is to actually get it finally published. So it's my great pleasure to formally be able to launch it. And Ishan, of course, had a major role to do with this. And I want to congratulate, I guess they're all 4 editors that had to work very hard to make this happen.
Bhavik, could you please make the presentation?
Sure. Can I have can I be made a presenter? Hello, Professor Bruce Alberts and Dr. Ishan and other participants. Can I ask someone to give me a remote control for so that I can show the presentation?
I think you can show it. Just try sharing the screen. It should work for you.
You share the screen now.
Okay, perfectly fine. I believe that everyone can see my screen now.
Not yet. Try that again.
Yeah. Is it working now, Dr. Said?
No.
Okay. Can you see the screen? So that means I haven't been given access yet.
No, if your part is green, the share screen is green, then I think you have access. Is it green at your end?
Nope.
Okay. Vaishali?
Sir, we've given the access to sir. He's already a co-host for the meeting, so he should have the access. So can you
Okay. Is it working
Yeah,
now?
it's coming
Yeah,
now.
it
Yes,
is working now.
it's there. Thank you. Just make it
Okay.
full screen.
Thank you.
Perfect. Yeah, perfectly fine. Give me a second, please. Okay. So it's my uttermost pleasure that I'm going to present the book launch of the important book and very potential book on biology of glial cells, recent advances. Now, along with Professor Bruce Alberts, I will inaugurate this book virtually, and so telling something about this book. This important book, by eminent professors and scientists and stalwarts in this field, reveals the important role of glial cells in neuronal health and disease. Moreover, it examines the structure, function, and maturation of glial cells. Moreover, it discusses the latest understanding of glial cell differentiation and homeostasis. It plays an important role in understanding the role of oligodendrocytes in myelination and demyelation. Further, it examines the function of astrocytes in healthy central nervous system and also reviews the role of myeloid cells, inflammation, and brain health. It is edited by the stalwarts in this field, and the editors of this book are Dr. Ishan Patro, who is the professor of zoology and neurosciences at Shivaji University, Gwalior, and the former vice chancellor of Raman Shah University. The second editor is Dr. Pankaj Seth, who is a senior professor at National Brain Research Center, which is an institute of excellence in neurosciences in India. The third editor is Dr. Nisha Patro, who is working as a scientist in the School of Studies in Neuroscience at Shivaji University, Gwalior. And most importantly, it is edited by also Dr. Prakash Narayan Tandon, who is a national research professor and the founder president of National Brain Research Society, Manesa. This book is under production and will be published in the January 2022. And this provides a complete understanding of role of glial cells in in central nervous system. With this, I thank the editors for their splendid work in putting up a great book. Thanks a lot.
Thank you, Dr. Bhavik.
Yeah.
May I now request Professor Raju to present the honorary fellowship to Professor Bruce Alberts. Professor Raju, please.
What a great honor indeed for the Academy to present the fellowship to Professor Bruce Alberts. Professor Bruce Alberts, you are now a fellow of the Indian Academy of Neuroscience. Congratulations.
Thank you very much. It's a great honor.
It's now my duty to thank Professor Bruce Alberts for sparing his time and talking to all of us. And I'm so happy the large number of students, scholars, and faculty members all over the country who are listening to your lecture through both Zoom and the YouTube. And possibly we'll be getting many compliments and queries, which we will communicate to you in subsequently. Over to Hova, please.
Thank you, sir. We would like to extend our heartfelt gratitude to Professor Alberts. Thank you, sir, for the enlightening talk and sharing with us your experiences in life. So we are really honored to have you with us in this event. Thank you very much. Now, it's my great honor to welcome and introduce our distinguished guest, Dr. Prakash N. Tandon, to this morning's inaugural ceremony. Dr. Tandon is the founder president of National Brain Research Center, Manesar. He is the father figure of neurosciences and has made immense contribution to the growth of neuroscience in the country. He is fellow of all the national academies and was the president of the Indian National Science Academy. He has been awarded Bhatnagar Fellow by CSIR, Padma Shri and Padma Bhushan by the Government of India. Thank you, sir, for accepting our invitation and a very warm welcome to you. I would now request you to kindly address the meeting.
Thank you very much for giving me this opportunity to meet old friends. It is unfortunate that we are on network system and not meeting personally, which I would have loved to do so. I must thank the president and the members of the executive committee for giving me this opportunity. I must say that I have been involved with this academy from very beginning. It was founded by my closest friends like Dr. K.P. Bhargava, Mehdi Hassan, Parmar, Dr. B.N. Dhawan. Unfortunately, some of them are no more. I take this opportunity also to thank my very dear friend, Professor Bruce Alberts. Bruce, to me anyway, is a great Indophile. As we have just heard, he has visited this country 20 times. And equally, credit should go to Betty, who many times forces him to come to India, even if he is not very willing to do so. Bruce has contributed a great deal to Indian science, and Betty to promote Indian culture, both in this country and especially in US. If you visit her home, the whole house is cluttered, I'm sorry to use that word, with Indian art and artifacts. That's the degree of their commitment to us. And as you have known, that we have known each other for just a little under 30 years. Some of you were still in your kindergarten at that time. But you should know that this is how science develops, not in silos, but in cooperation with people all around the world. And he has done a great deal in this respect when he was the president of the National Academy of Sciences. Let me now share with you some thoughts about the evolution of research on glia in this country. These days, for the last 2 years, I have been busy tracing the evolution of neuroscience over the centuries, mostly abroad and to some extent in India. And therefore, it is my pleasure to talk about the evolution of research on glia in this country. A great credit for this should go to your president, Ishan. It was, you see, by the time I finished my professional career in 1990, and I take credit that I kept myself up to date in my knowledge as far as possible, there was very little I knew about glia, in spite of my efforts to be up to date in most of the current knowledge. So what did I know in 1990 about glia? I knew the role of astrocytes in repair of brain injury, a subject of great interest to my mentor, one of my mentors, Dr. Violet Banfield, who in search of the etiopathogenesis of glial scar formation, which was responsible for epilepsy, traveled to Spain, to the lab of Santiago Cajal and Ortega. So that I knew a little bit. I knew a little bit about the oligodendroglial cells doing something to myelin. And then I learned much more about it, of course, because of my friend from Indian Institute of Science, who worked on this subject a great deal. I knew, of course, a little bit about microglia, that they are the resident immunocompetent cells in the brain, and therefore are very active whenever there is an insult to the brain. So that was practically all that I knew. In 1996, among one of the letters from Ishan from Newcastle, where he had gone as a fellow, he mentioned to me that he has taken up a study on role of astroglia in pathogenesis of Alzheimer's disease. That sounded a little unusual to me. After all, the Alzheimer's disease is because of the loss of neurons, and here he is studying astrocytes. Anyway, that aroused my interest. So I looked at some literature and got interested in the subject. When Ishan returned to India, in consultation with me, he applied for a research project to DVT. And that was not on astroglia, but microglia this time. The role of microglial activation in neuronal apoptosis. And another study on microglia in adult neurogenesis, because that was a very growing subject at that time, adult neurogenesis, which was not believed to happen till around that time. Anyway, he completed these studies, and we were very pleased. The kind of work that he had carried out was appreciated greatly by the neuroscience committee of DVT, and wanted him to promote this field in the country. And for that purpose, he organized 3 consecutive symposia on the subject of glial biology in Gwalior. I happened to attend the last one of them. somewhere around 2010. At that stage, Department of Biotechnology decided to promote this field further and asked Ishan to have a network of scientists working in this field in India on specific projects to promote this area. That was I think it was around 2012 that it was done. And at that time, searching for those who were working on glial biology in the country, he found only 13 groups that he could really involve in this task, and produced a report for the Department of Biotechnology. At that time, or around that time, I should say, your dean, Dr. Raju in Indian Institute, National Institute of Mental Health and Neuroscience, was also working, utilizing a model for evaluating the pathogenesis of ALS along with one of my other students, then director of the institute, Dr. Gauri Devi. There was also Pankaj in National Brain Research Center, with whom I had obviously to interact very frequently, being associated with the center. He was trying to develop a platform to generate brain cells. And that included, besides neurons, and modifying these neurons or neuronals within code neuronal cell into astrocytes and oligodendroglial cells. The purpose was, of course, basic research, but more important at that time was his interest in the field of biology of HIV, and to see the effect of the role of these cells in the pathogenesis of the neural involvement in HIV. When that was over, came the Zika cell problem in the country, and he used this platform, or the cells generated in this platform, to study the pathogenesis of brain damage in Zika encephalitis or Zika infection as such. And I'm told now that he's working on the same thing with regards to the present SARS virus, or the responsible for the COVID infection. So this prompted me to catalyze the development of this book. I thought it will be important to bring out a book summarizing Indian contributions in the field of glial biology, not only of relevance to Indian conditions, but also for its international knowledge advance. And you have, you will see this book in a month's time. Now, I have stated all this not to tell a story, but to tell a message that while starting neuroscience in this country, clinical or basic, what all we had to go through and the time span on which we developed. Thus, when we started neurosurgery in this country, which was started obviously by even my predecessors, that is Dr. Chandy, Dr. Ramamurthy, Dr. Ginde, in 1950. In 1962, when I attended the first meeting of the Neurology Society of India, we were a handful of people present there, neurologists, neurosurgeons, neuropathologists, and you will be, this will be of interest for you to know that when we used to meet for presentation, for our paper presentation in the conference, you could go on talking for as long as you wanted because there was all the time and very few people to talk. And from that, it has gone to the present state, 1950 to 2010 or 2020. More than 2,000 neurosurgeons and equally neurologists. The same thing happened with the basic neuroscientists. When the Academy of Neurosciences was started, there were really a handful of scientists who were working in the field. The first meeting that I attended of the Academy of Neurosciences, this was, I think, 1984 or 85. Dr. Seth can correct me, in Lucknow. There were only 70 persons who had registered for the meeting. And today he finds it difficult to have a meeting to provide for a platform for scientists so that the meeting is now this time, I'm told, for 4 days. I think this is creditable, and great credit for this should go to not only the founders, but those who have continued to follow the development of this field. So let me close by saying, Der aaye durust aaye. We started late, but we have come to a stage when our work is meaningful. And therefore, I take this opportunity to wish you Godspeed for future progress. Thank you very much. I think I should close here and pass it back to the convener.
Thank you very much, sir, for sharing with us your experiences. We also have with us today a very senior professor, Dr.